I'm Janet Rowley. I'm a professor of medicine, molecular genetics and cell biology, and human genetics at the University of Chicago. I've been on the faculty of the University of Chicago since 1962.
I'm Al Knudson. I'm a senior member at the Fox Chase Cancer Center in human genetics, and I've worked there for 27 years. Janet, I'm very interested in knowing more about the beginning of your life and how you did or did not have an interest in science, even as a child.
Well, I'm truly a University of Chicago product because both of my parents went to college at the University of Chicago in the late 1919, 1920, so that there's always been a close affiliation and affection for the University of Chicago. My mother was a school teacher. My father was in retail store management and then a professor. So that it's been part of my life that, and I'm an only child, so my parents expected me to do something that they thought would be special. And they encouraged me in that. It's interesting that I came to the university actually as a junior in high school because the university then was organized along a different pattern, and Robert Maynard Hutchins thought the college should really start in the last 2 years of high school. They offered scholarships to come to the university, which my mother as a teacher knew about, and so I applied for a scholarship, won one, and that was the beginning.
You really had imprinting of academia. You obviously didn't pursue the precise things they did. When did you start deviating from the path that they set?
I went to a Catholic girls' high school for the first 2 years, and I had a fabulous nun who was a teacher of zoology. And she taught zoology in a way that was, I'm sure, is unique because she went into all of the phylogenetic trees, and we had to learn the precise criteria for different orders, different phyla, etc. And it appealed to me, the sense of order and the logic I thought was fascinating. But when I came to the university in the four-year college, I actually was most turned on as a freshman in college with a general biology class in physiology. So I registered then, when one had to pick a major, I registered as a physiology student. I think that luck plays an important role in how one's life develops. And in this context, as a physiology major, I was taking classes with pre-med students. And so we were all together dissecting cats and doing all of the other things in comparative anatomy. And finally, I decided that I really was more interested in people, and medicine is what I chose.
You were getting into pattern recognition, too.
I did that earlier. My father was a stamp collector. And so when I was probably about 9 or 10 years old, he gave me a bunch of stamps and a little book to put them in, an album, stamp album. And I thought it was fascinating to try and figure out stamps. And you talk about pattern recognition in American stamps back in the late 1890s, 1900s. The difference between stamps as identified as unique stamps in a stamp catalog are little watermarks. So you really have to look at very fine detail in order to distinguish one stamp from another. So I was in the pattern recognition very early.
So on the one hand, you had all this classification, and on the other hand, you had the way things work.
Well, that's right.
So when you got to medical school, was it what you thought it would be?
Getting into medical school back in 1944 was a challenge in itself. And the university had a class of 65 students, 3 of which were women. And so I applied for the class to enter in 1944, and the quota for women was filled. Now that isn't such a tragedy because I was only 19 years old and a pretty naive 19-year-old at that. So I then went in the next class, which was 1945. By that time it was clear the Second World War was winding down. The Army Navy didn't need so many slots in medical schools, so suddenly they opened it up to 4 Fs and women. And so we had 7 women in a class of 65 when I entered.
So did you have any women models in medical school?
I had a number of women, and I think, again, I look on myself as being very fortunate being at the University of Chicago because women have played an important role as professors, as administrators from the time the school was founded. But there were very senior women, both in the Department of Surgery, Department of Medicine, Pathology. When I was a medical student, a few of them were married, but most of them were unmarried, and it was medicine was their whole
They were
whole
married
life.
to medicine. How did you see the basic sciences versus the clinical?
When I was going through medical school, I didn't consider basic science or say an MD, a PhD as an option. Though again, the university was unique in that about 20% of our class were individuals with PhDs, so they had a very strong research tilt.
So when you were in your last year, let's say, of medical school, what did you think you were going to be doing?
Well, I have to confess that the last 9 months of medical school for me was thinking about getting married because I had become engaged, and in deference to my mother, who was afraid that if I got married before I graduated that somehow I wouldn't graduate, we postponed the wedding till the day after medical school. So that then changed things because my husband, and we've been married more than 55 years, my husband was also he had got a master's degree in pathology, but then was starting back in medical school. So I decided I would wait, and the two of us would intern together, which meant that I did some other things for a year and a half.
You didn't have some big long-range plan.
I had no long-range plan at all. In retrospect, probably turns out to be a good thing. My husband went to NIH, and I obviously went along. So this was after a one-year internship, and he started a research laboratory at NIH. And I then worked in clinics baby clinics in Montgomery County and prenatal clinics there. And I could do that 2 or 3 afternoons a week because within 9 months of the time of arriving at NIH, I had our first child. So I wanted to spend most of my time at home with the baby.
So you were never completely away from medicine?
No, but practicing in a way that was compatible with, having some medical practice, but at the same time being able to spend a lot of time with the young baby. But I had lots of other things I wanted to do, and we just bought a new home that was plain clay, and I had to make a garden, and so I had other things to occupy myself.
Was that part of the Mendelian influence that you were a gardener?
More because I was the daughter and granddaughter of British gardeners, and so that was what they liked, and I was had grown up with gardens.
You and I both had the experience of going through medical school without hearing about genetics at all when we were students. It was like the subject didn't exist for medicine. Is that pretty much true for you?
That certainly is true.
Yeah.
I think it came through most clearly actually in neurology clinics, because there a number of the neurologic disorders are genetic, and particularly in pediatric neurology, and so you get a smattering of it, but certainly nothing in a formal sense. Genetics was not taught in medical school because it did involve rare patients, and so it was only when you got out of medical school and became a resident and working in specialty areas that you would see many of these patients. The whole notion that genetics was really about eugenics and forced sterilization of individuals made physicians very leery of having much to do with that as a discipline.
So then you did wander and travel.
My husband, again, having had returned from NIH to Chicago and having a position at the university, took a sabbatical at the appropriate time, and we went to Oxford, and I was casting about as what I could do. I had had the good fortune toward the end of the late 50s to be working in a clinic for retarded children, and then Down syndrome was discovered to be trisomy 21, and of course in the clinic for retarded children many of the patients had Down syndrome. So it seemed logical, since Oxford was a center for chromosome research, for me to apply for an NIH fellowship to learn cytogenetics, which I got, and be accepted in a lab in Oxford, Laszlo Leida's lab, and so that's when I began to study chromosomes. Now, Laszlo didn't know anything about chromosomes. He was really interested in DNA replication in hematopoietic cells, in bone marrow cells. But down the street was an MRC unit, and Marco Ficcaro, who's now at Pavia, was there. And so Marco taught me cytogenetics. The second sabbatical that we took in Oxford was based on Casperson's work and the study of replication, or the pattern of DNA in plant chromosomes when stained with quinacridone mustard, that banding was identified.
Mm-hmm. When one looks back on the time from, let's say, the discovery that the human chromosome number was 46 instead of 48, until the time of quinacridone and banding, was a pretty short time.
1956 to 1970. Yes.
So you were right there all that time.
Well, I certainly had a ringside seat on much of what's developed.
Yeah. When did you start calling yourself a cytogeneticist?
Well, probably fairly soon after I returned to the United States. So after we finished the first sabbatical in Oxford, I decided that the research project that I had started on, which was looking at the pattern of DNA replication of chromosomes. We focused on the X chromosome because, of course, it's one of the X's replicates very, very late. But at that time, you couldn't prove that it was an X chromosome because all of these chromosomes had relatively similar size and shape. Yep. Marco Ficcaro was a close friend and colleague of Jan Lindsten, and he was at the Karolinska Institute in Stockholm. So Marco made arrangements for me to go to Sweden and get peripheral blood on a number of Jan Lindsten's patients. And I went there with tritiated thymidine. I'm sure you can't even carry radioactive material now, but I had a little vial that I carried from Oxford to Stockholm, added it to cultures from cells with patients with many different kinds of X chromosome abnormalities, and then brought the material back to Oxford to work on. I had not much time in Oxford. We did publish one paper in Nature on a patient with 4 X's and a Y, a male, and showed that 3 of the X's were late labeling, so that this further confirmed the notion that the late labeling chromosome was the X. But I came back, of course, with my husband and family in '62 and decided that I was going to continue to work part-time, and I had to make a choice, either returning to the Clinic for Retarded Children or continuing with the research. And I found the research fun and challenging, so I approached one of my professors, Leon Jacobson, who was then chairman of the Department of Medicine, but also director of an institute that was funded by the Atomic Energy Commission. You know, I asked him, can I come and work? And I need some bench space in the laboratory. I need use of a microscope and a dark room. And by the way, would you pay me? And Dr. Jacobson said yes to all of those things. I had no credentials whatever, like paper in Nature, but you know, that's not much. No training, but he was a very generous individual, and it all started then. He was central to it because nobody at the university knew what I was doing, and yet they allowed me to continue. I think it's important to say that this institute that Dr. Jacobson directed had really a block grant. So I didn't have any grants in my own name for the first 10 years of my research. And the other reason that Dr. Jacobson is important is that he was a hematologist. Yep. So he was taking care of patients who had leukemia, and he was particularly interested in patients who had what he and some of his colleagues here at Chicago called pre-leukemia, which were patients with unexplained low white counts or platelet counts anemia. And so I began to do chromosome studies on his patients and found that they had chromosome abnormalities. The problem in 1969 when I was doing this work was we couldn't tell one chromosome from another within large morphologically similar categories. And these patients had gains or losses of what was then called a C group chromosome. But you couldn't tell from, well, that's right, 16 chromosomes in females, and you couldn't tell what, so 2 patients who lost a C group chromosome, was it the same chromosome? Was it different in the 2 patients? One just didn't know. And this then brings up really the work of Peter Knoll, because so I was doing this work in the middle 60s to the late 60s, and of course Peter and David Hungerford had identified the Philadelphia chromosome in CML as a unique marker that was related to this kind of leukemia. But that was really an aberration when you looked at most cancers. And it's important to realize that at that time, most cancers that were studied were from animals. Most of them were ascites, that is, fluid that develops in the lung, easy to obtain, lots of cells, no taking solid tumors and cutting them up and things like that. And so you had the most aberrant cancer cells that were being studied, and they were looked on as typical.
All this time you were at the University of Chicago. Now I remember Beadle left Caltech around late 50s to 60, something like that. Did you have any interaction with him all this time that you were developing your career?
I had not much interaction with George Beadle until the middle 60s. He was instrumental in inviting the organizers of the International Congress of Human Genetics to come to Chicago and have their international meeting, which occurs every 5 years. And the first one had been in '61 in Rome. So he invited them for the second international meeting in '66 here in Chicago. And I got to meet him at that time. And then we developed a friendship such that when Mr. Beadle moved from the president's house, he came and told me that they were going to be digging up his back garden and just paving it over for cars for the next president. He had a lot of daylilies, and what I like, some of his daylilies. So he brought over several bushel baskets of daylilies to the garden.
We mentioned your being in Oxford, and of course you met some great intellects there. And I know that one of them was Walter Bodmer of genetics fame, now known a lot for his interest in cancer, but at that time he had not yet worked in that field, to my knowledge. Did you have some contact with him, or learn from him, or did you influence him?
Well, potentially, though I think you have to realize that I went to Oxford the second time in 1970, and that's just when Walter and Julia were coming from Stanford to Oxford, where he'd been appointed the first professor of genetics. He was just starting his unit, so the first 6 months it was a matter of the whole laboratory being under construction and very few people having lab facilities there at the time. One of the people that Walter brought from Stanford with him was Marcus Nabholz, who was Swiss and who's back in Switzerland. But Marcus was working on somatic cell hybrids, and that really related to Walter's interest in genetics and trying to do mapping of genes. And you remember Boris Afrusi and Mary Weiss,
absolutely.
The first ones using somatic cell hybrids to show that thymidine kinase was on chromosome 17, because when you selected in cells that lacked thymidine kinase, chromosome 17 was lost. So Walter was doing somatic cell hybrids with mouse human cells, mainly mouse 3T3 cells, which have a very wild karyotype. He was interested in someone looking at the chromosomes of those mice. I actually set up Walter's cytogenetics laboratory for him there at the Genetics Institute and published the first paper out of the laboratory, which was showing the pattern of quinacridone mustard bands with and quinacridone mustard being a dye that stains human chromosomes. It's interesting that while the laboratory was under renovation, I actually then returned to the place where I had worked part of the time, the first time in Oxford, namely the Medical Research Council unit, Population Genetics, that Alan Stevenson was the director of. This time Peter Pearson was there, and Peter was very early into using quinacridone mustard stains to look both at human chromosomes and the pattern of banding in human chromosomes, but he was interested in the great apes and in comparing the pattern over evolution between great apes and humans. So Peter taught me banding, and we happened to live near the MRC unit so that I could go in nights and weekends and use his microscope to look at Walter's cells, because we didn't have the facilities at the in the new genetics unit to do that, or at least not for the first 6 months. In the meantime, and I referred to this earlier, having studied patients with these pre-leukemic conditions with chromosome abnormalities that I couldn't identify, I sent back to the United States for them to send me slides so that I could start and do the new quinacridone banding on the patients to begin to define the identity of the chromosome abnormalities that I'd identified as general abnormalities, but now I could tell precisely what chromosome was missing.
So Peter Knoll and Hungerford published this paper, which was extremely important because it was the first time a specific genetic abnormality was associated with a specific cancer. And nobody understood what was happening until you came along. So this was a great discovery. The question is, why didn't people pay more attention to Nolan Hungerford?
Because most cancers have such wild karyotypes or karyotype being the pattern of chromosomes when you actually analyze them in detail. There's so many gains of chromosomes, many more than the 46 that normal cells have, many odd shapes, including ring chromosomes, so that the fact that there was this one example in a relatively, leukemias are rare, so in this one rare situation, most people just said this is the exception that in fact proves the rule that by and large cancer is associated with many chromosome abnormalities, that they appear to be different from one cancer to another, even the same kind of cancer. And so the notion in the 60s and early 70s was that chromosome abnormalities were a result of genomic instability in cancer cells, and they had no role at all in the actual development of those cancers. So that when I made the discovery first in an acute leukemia of a translocation involving chromosome 821, and then the second one just 6 months later, looking at patients with the Philadelphia chromosome, showing that that really was a translocation involving chromosome 9 and 22. Now, people ask me the question, about why I was looking for translocations. That's not the question at all. Translocations just happen to be the mechanism. And I actually was looking at patients with chronic myelogenous leukemia, or CML, in what's the advanced stage or the acute terminal stage of leukemia when they have other chromosome abnormalities. And I was really interested in what were the identity of the chromosomes that were present as extra chromosomes. Again, they're primarily these chromosomes in this large C group, and so you'd have several extra ones. Were they the same? Were they different? It turned out that the extra chromosomes were mainly chromosome 8, and so they were consistently the same. And then patients had other abnormalities as well. So this was a time when I was still working part-time. And you take photographs of chromosomes, and then I would take all the photographs home and cut out the chromosomes individually, put them on my dining room table, and just try to arrange them according to the proper pairs, focusing really on what was the identity of all these obviously abnormal chromosomes. And in the process of that, it became apparent in the first 2 patients that I looked at that one chromosome 9 was abnormal. So they clearly had the Philadelphia chromosome, but here 9 was much longer than the other with pale material. And you sort of look at that and say, this looks like a translocation, but I had material from these same patients in their earlier chronic phase when they just had 46 chromosomes with the Philadelphia chromosome. And those very early cells had the translocation already showing 9 and 22. So this wasn't just an additional change in the acute final stage. It really was there at the earliest stage that you could study the patients. And having already seen one translocation, I thought, wow, this really is a translocation, and it's important. So I wrote the paper to Nature and sent it off. And again, this is sort of part of education. When I worked with Walter and in Oxford, and we did the paper showing the chromosome pattern and the comparison of 2 different staining methods, Walter sent the paper to Nature, and it was accepted and published in 6 weeks. So I thought, well, this is even more important than the paper I wrote with Walter. They're going to publish it right away, and it took them 6 months. Firstly, it was rejected, and then accepted in 6 months before it was published. And it's a relatively simple thing to stain the chromosomes and look. And I had been talking about this, and I fully expected to find somebody else publishing the same data as I had discovered, but fortunately that wasn't the case. And I want to make one other point here that in a sense is off the subject, but it is that I made the discovery of the Philadelphia chromosome when I was 48 years old. And so that's really the first important scientific discovery, really important discovery I had made. And it seems to me too many people, particularly young people, look on biology as equivalent to mathematics, and if you haven't done something that's noteworthy by the time you're 19 or 21 or whatever, you should pack up and go home because you've got a bleak future. And I think that it's possible to do important things working part-time and at a later stage in your life.
My paper on 2 hits was when I was 48.
A good age, a good age.
How long did it take before it was really accepted and understood? Did it require molecular cloning of the genes before it was really accepted, or was it pretty much accepted without that?
You know, it actually came in 2 stages, and I was fortunate. So the 821 was 1972, and the 922 was published in 73, and then in 77 I discovered the 1517 translocation in acute promyelocytic leukemia. And the fact that each one of these was specifically associated with a particular type of leukemia convinced me that chromosome translocations were critically important. And Laura Zek, I believe it's 1984, discovered that Burkitt lymphoma was an 814 translocation. So it wasn't just this rare form of leukemia, it was also found in other things. Also the fact that other chromosome abnormalities, gains and losses of chromosomes, were consistent and not entirely random. I was convinced that chromosome abnormalities were important, and I would go to national meetings of the American Society of Hematology. The meetings, the education sessions for neoplasia are held on Sunday mornings, and I would get up there and tell people that they ought to be in church. But since they weren't in church, they were going to get their sermon from me. And the sermon was the chromosome abnormalities were important, and they had to begin to pay attention to them. And as time went on, it became apparent that chromosome abnormalities are the most important prognostic features for some of the acute leukemias, such that if physicians knew the karyotype, the chromosome pattern of the leukemic cells of patients, they could predict which ones were going to do well and which ones would not do well. So that it was this combination. But I think that it wasn't until the genes at the translocation breakpoints were identified that it really became more widely accepted, and that certainly the papers in 1982 from Phil Leader's group and Carlo Croce, Riccardo Della Favara, collaborating with Bob Gallo, 2 papers in the same issue of PNAS in November of '82, that really convinced people. And it's interesting, it convinced people in both ways. There were some people that said that they finally believed that oncogenes were important because they were involved in these recurring chromosome translocations, and others that because the translocations involved oncogenes, they believed the cytogenetics. So we all benefited. Some people were more easily persuaded,
especially
I think. Especially
geneticists.
Yes, more easily persuaded than others. But you know, it's interesting to think back to those times, and I started going around and talking to people about how can you explain consistent translocations? Why they should be these 2 chromosomes wasn't clear, and it was only clear on cloning the breakpoints, and those were events in the 80s.
The discovery of Bishop and Varmus and that group, that there are proto-oncogenes inside us, together with the later finding of Neal and Hayward and Astron about promoter insertion of viruses that don't carry their own oncogene. These things, I know that they attracted your attention.
Well, particularly when the genes that, at the translocation breakpoints were identified. The first one being cloned, of course, was the E14 translocation in Burkitt lymphoma, and that involved the MYC gene on chromosome 8 and the immunoglobulin gene on chromosome 14, so that then MYC became front and center, if you will. It is interesting in that regard that just recently, as part of gene therapy experiments for an immunodeficiency disease, that the vector that's used integrated in a particular gene called the LMO2 gene and caused leukemia, T-cell leukemia, in 2 of the patients who've been transplanted. And the results there reminded me immediately of Susan Astron's work, because this was an insertion of the virus adjacent to a gene that was first identified by cloning a translocation breakpoint and showing that viral insertion happens in individuals and can lead to serious problems. Fortunately, one of the patients has had a complete remission and is doing very well, and his immunologic disease is better.
Then subsequently there were other supporting things too with the DNA viruses.
I mean, this leads into the gene that's involved in chronic myeloid leukemia, or CML.
Yep.
And the 922 translocation involves the Abelson gene on chromosome 9 with a gene that was identified when the breakpoint was cloned, the BCR gene. But the Abelson gene was discovered because a virus picked up a part of the Abelson gene and caused leukemia.
And it's created this enormous excitement in recent times because there's a drug that works specifically against the action of that activated oncogene. This must be really wonderful for you to see your gene be the source of the first directed therapy in cancer.
Well, it is very rewarding. It's the only way to put it. And this is the kind of thing that myself and other individuals hoped would happen by leading to this greater understanding of what's really going on. I think that there are several important things one can emphasize here. Firstly, that's almost 40 years from the time that Nolan Hungerford discovered the Philadelphia chromosome in 1960 to the translocation 72-73 to cloning in 1984 and identifying the genes, and then finally the development and approval of, development of the drug leading to trials in 1998. And that also involved efforts along the way, and not necessarily coordinated efforts, but efforts that turned out to be collaborative between cytogeneticists and molecular geneticists, and then chemists, because CIBA is the company originally that identified the tyrosine kinase that inhibits the Abelson gene, and then Brian Drucker at Oregon and Charles Sawyer at UCLA, who together started clinical trials showing that it was effective in the individuals. So this is really great, and of course Novartis is now making a great deal of money. It has another side to it in that most people have said you can't get specific drug therapy for leukemias because they're rare and drug companies only want drugs that are going to treat hundreds of thousands of patients. It's something like 10,000 patients a year get leukemia or subtypes of leukemia, and 200,000 get breast cancer or lung cancer. So, leukemias are orphan diseases, if you will, in that sense. So the fact that Novartis can be financially successful with a targeted therapy targeted to a small population gives me hope that maybe all the other translocations that we've identified will themselves become the focus of research.
You decided to move your lab into the molecular era. Can you tell us about that and how it came about and how you made the decision to do that?
Well, it was very clear to me, even before translocation breakpoints were cloned, that with increased understanding of genes and where genes were located on chromosomes, thinking this is back in 1980, that there ought to be some way to figure out more about the genetics of the Philadelphia chromosome. But clearly, as a cytogeneticist, I was not in a position to do that. Very fortunately, I had the support of the head of the section, because I'm in the section of hematology oncology in the Department of Medicine, and the section head was Dr. Harvey Gollum. And Harvey was able to get some additional laboratory space in hematology oncology, and he agreed to give that space to me. And then also that he would help to finance the recruitment of someone to come with the kind of training that we needed to head that lab. And I was fortunate in recruiting Dr. Manuel Diaz, who was working with Joe Gall and working then on salamander chromosomes. So he came in 1982, which was just when translocation breakpoints were cloned. And we began by trying to figure out some of the genes that might be abnormal or abnormally expressed. I was always interested in the fact that there were many different translocations involving chromosome 11, a particular band on the long arm, which is numbered 11q23, and that both leukemias that involve bone marrow cells, the myeloid cells, as well as leukemias that affected the lymphocytes and lymph nodes, both had translocations involving that region of chromosome 11. And was it the same gene or not? Oh. Michel Lebeau then used new techniques in cytogenetics to work with Manuel in mapping genes that could be involved. So we were doing fluorescence in situ hybridization, where you would take DNA and label it with probes that glowed in the dark, or glowed in ultraviolet light, so-called FISH. And in collaboration with Maynard Olson, we got probes from that region of chromosome 11 and found one probe that was split. So we knew that we had within this single probe, which was over a megabase, over 1 million base pairs in length, but we knew that we had, at least we were getting close to the gene. And it was split in both leukemias that affected the bone marrow cells as well as that affected the lymphoid cells. So this was a paper that we published in 1990, and we called the gene MLL for myeloid lymphoid leukemia. And it's now, of course, the gene has almost developed a life of its own, with many, many different laboratories studying it from a whole large number of perspectives.
There's this story about so many partners. What do you make of that? And are some of these partners turning it toward lymphoid or toward myeloid?
Well, this is an important question, and actually MLL does have at least 60 partners that are involved in translocation. So that's 60 different genes. We should back up a bit, because in this instance, the translocation involves chromosome 11 and some other chromosome or chromosome region, and most of the chromosomes that are present in the genome are actually involved in translocations with MLL. They form a fusion messenger RNA, a fusion protein, which is unique for each different translocation. But many of them contain regions that should activate the MLL gene. And it's important, I think, to note that the MLL gene is a homologue of the Drosophila gene trithorax. So one of the other names for MLL, because many genes have multiple names, is HTRX for human trithorax gene. But why it has so many partners, we don't understand. The part of MLL that's present in all of these different translocations is virtually identical, give or take a few small exons whose function we're not very clear on. But there are certain translocations that are seen almost exclusively in the myeloid or the bone marrow type leukemias, and other translocations that are seen almost exclusively in lymphoid leukemias. So that would lead one to think that, as you suggested, there's something about the function of the partner gene that is influencing the MLL gene.
There are a couple of things of great interest. One is that a lot of these are in infants, and infant leukemia has always been a mystery. And secondly, that some of them are occurring after chemotherapy, cancer chemotherapy especially. And this form of leukemia arises and shows this 11q23 MLL gene. So would you comment about those two aspects? And have I chosen injudiciously two, and there are a lot of other aspects?
Well, those are two areas that have been of special interest to the laboratory, and we've approached this from several different ways. One of which is just to say, where in the gene of the MLL gene do the breakpoints occur in translocations? And it's interesting, the gene itself is about 100 kilobases long, but the breakpoints, virtually all of them occur within an 8.3 kb segment of that 100 kilobases. So it's not occurring all over randomly. It's in a very precise area. The breakpoints in infant leukemia and treatment-related leukemia occur in the back half, if you will, of this breakpoint cluster region, where the ones that occur in adults occur in the beginning half of this region. Why should they all be breaking in this particular region? Now, I think one important answer, but it doesn't get to the mechanism of breakage, is that if a translocation doesn't give a hematopoietic cell, a bone marrow cell, a blood cell, a proliferative advantage, you're not going to see that. So my view is that most breaks and rearrangements are probably either lethal or they're neutral, and you don't see them. All we see are those that lead to leukemia, because that's the population we select to study. But then, so we've then looked, the whole gene has been sequenced. There are some repetitive sequences called ALU sequences, and they particularly cluster in the beginning part of the gene, and actually not many, but some of the breakpoints in adult leukemia actually fall within the ALU sequence, these repetitive sequences. But particularly related to the second subset of patients, the treatment-related patients, we've also mapped a single site in the back portion of this region that is preferentially cleaved when you expose the cells to topoisomerase II inhibitors. So This is a topo 2 cleavage site, and there's only one in the whole gene. The treatment that is given to patients who develop this kind of leukemia almost always includes drugs that target topoisomerase 2. So our notion is that the drugs that the patients receive actually inhibit the function of topoisomerase 2, which ligates broken pieces of DNA, so it inhibits this joining together of DNA, and you're left with broken ends, and that this could be one part of the translocation.
Does the fact that some of these are caused by a known chemotherapeutic agent make you think that perhaps some of the spontaneous ones in that same region are caused by some environmental agent?
Some of the bioflavonoids that are high in fruits and soy products actually cut the DNA of this gene as effectively as the drugs that we're using to treat cancer. That raises the question if mothers, for example, who are pregnant during the early part of the pregnancy eat a lot of these soy products, might they be putting their unborn child at risk of developing leukemia? No. This is a possibility. I think without more data, one certainly would not want to encourage anybody to change their diet because of this, but it is a possibility. You raise, to change it a little bit from MLL, raise the whole question of what really does cause chromosome translocations. And I've indicated a certain bias to say that at least at the end, after the breaks occur, that selection plays an enormous role. We have evidence in some of the lymphoid leukemias and lymphomas where translocations occur that in fact the recombinase system, which is a system that involves, is important in the immune cells in rearranging the immunoglobulin receptors as well as the T cell receptors to make them mature functional genes. And they recognize certain DNA sequences. These same DNA sequences are adjacent to other genes which are involved in translocations in B cell and T cell leukemias. But I come back, if that rearrangement didn't lead to a growth advantage of those cells, you wouldn't, they wouldn't be seen as a clonal outgrowth in these leukemias and lymphomas. There's no evidence that the recombinase system functions in myeloid cells, in the bone marrow derived cells.
It seems that there's something remarkable that leukemias and lymphomas and a few sarcomas typically or most frequently involve such translocations. If we look at other cancers, we don't see that. And we rarely see point mutations in oncogenes. Is that due to the fact that you can get much higher activity out of your oncogene with a translocation than you can with a point mutation? And why is it related to these particular tissues?
Well, that's an important question. The requirement for the translocation is that it's very specific. It's a precise rearrangement. Yeah. If you don't lead to a protein that's a functional protein, that translocation is not functionally relevant. And I look on the genetic changes in solid tumors as being much more permissive and, if you will, allowing a lot of sloppy things to happen to get rid of a tumor suppressor gene. It's pretty clear that a translocation is not in and of itself sufficient to get a leukemia. That you have to have the translocation plus other genetic events to really have a full-blown leukemia or lymphoma. And what those other events are, we basically do not know. And we have some preferred genomic regions that we think are going to be important because many of the translocations have other chromosome abnormalities associated with them quite frequently, deletions, for example. So those deleted regions should be the area of oncogenes. But probably except for the deletion on the short arm of chromosome 9, where a number of genes, INC4A and others related to the cell cycle are located, we don't know the identity of any of those second hit genes, if you will.
So here we are, we're 2004, and do we have enough understanding to really go about rational therapy and prevention?
Well, this reminds me of the kind of discussions we used to have when I was on the National Cancer Advisory Board, which is the council for the National Cancer Institute. And there were many people who wanted to spend most of the money on clinical trials and treatment. And there were others of us who said we don't understand enough to really treat people effectively. And this was back 1979 to 1984. I think we're still not exactly in the same state as we were 20 years ago. We've made substantial progress, but I think we still underestimate the complexity of the problem of cancer and our relative ignorance in that regard. So I think we need to learn more to really be able to do effective treatment and probably effective prevention. I think we have enough understanding in some areas to move into rational therapy, and clearly not only Gleevec, but the fact that we use all trans-retinoic acid to treat acute promyelocytic leukemia with the 1517 translocation. Those are evidence of past successes, and we need specific therapy for some of the others. I think it's going to be much more complicated because there are all sorts of areas that we don't know about. The role of RNA in regulating gene expression in normal cells, let alone in cancer cells, is a virgin area as far as I'm concerned. So and that's just one example of things that we need to know.
Janet, earlier on, we brought up the fact that in medical schools, women were a really profound minority. Did being a female matter much in your career?
Well, I think I was very fortunate in being at the University of Chicago, and this is a theme that's come through several times, in that in general, it's far more welcoming of women than many institutions. I think that there are several problems today facing women. As I've indicated, when I graduated from medical school, I treated medicine as a hobby. So my family came first, and I worked part-time in clinics. I worked part-time until my youngest of 4 sons was 12 years old, because I thought that it was important for me to be home, and also I had lots of other interests that I wanted to be able to pursue. That's not the way women look on themselves and their careers these days. So I think they're much more demanding of themselves and their professional advancement than I was. And this is a difficult problem. And if you want to be a woman taking care of your children and also be at the top, that's an extremely difficult challenge for women. And I don't think that particularly academic institutions do a good job of trying to allow women to have a satisfying life as a mother and also helping and supporting them during their career development.
One of the things I see, and maybe I'd like to know your view, is that we count people's papers and the total productivity without placing value judgments on what's being done.
I think that is true. It's the number of papers or the weight of papers that's driving things, and not necessarily individual creativity. But in order to be really creative and think of things that are far out, part of that comes with just the way you think, but it also is that you have time to contemplate and to evaluate what it is you're wanting to do.
So you've had a life of service as well as your own science. Is there a temptation on the part of good scientists not to participate in service so they'll get more work done? Should there be a sense of obligation that the very good scientist participate more because he's a very good scientist?
I would certainly support that. Because you have to have people who have some perspective in order to be able to make choices amongst the different opportunities. And I'm on the council now of the Human Genome Research Institute. And one of the things we're discussing now is what proportion of funds for the Genome Institute should go for both patient care as well as general biology. And we should take some of the resources that went into sequencing and use them now in these other areas. But that requires judgment, and judgment of many different individuals discussing it and coming to some kind of consensus.
Are you suggesting that we should have more thought and discussion about the future and how to get there?
Oh, absolutely. I think that's certainly on both the national level as well as in academic institutions. It's important. We've lived through a golden age in biology, but I think there's going to be another golden age in the next 10 or 20 years. Many of the major questions that we have in biology are only going to be answered in collaboration with colleagues in the physical sciences as well as mathematics and computers to really understand at a precise level how proteins interact, how energy is exchanged in cells, how the mutations that we've talked about really do occur. What is a break in DNA? What does it mean to rejoin two pieces of DNA that may be at some distance in a cell, but obviously when they rejoin, they're together? Those are questions, some of which haven't been asked except in a very general sense, and I think those are the things that we need to focus on in the future. Because if you just look at chromosomes, firstly, 1956 was when we discovered that humans have 46, not 48 chromosomes. That was a technical advance. We then could look at the chromosomes and sort them by size and shape, but we couldn't tell them one from another. Banding in 1970 transformed a whole area. Now being able to use DNA probes that are labeled with fluorochromes that glow in the dark and see where the DNA is, how it moves, all of the things we can do much more precisely with smaller and smaller DNA probes has given us great insights. This is what's going to happen in all of biology in the future. Cancer, but all areas of biology. So I think the future is really going to be glorious for all people in many different disciplines, and it's going to be because we can answer questions that are ever more detailed questions, and they'll lead us to new understandings and insights.
So here we are. You've had a wonderful career in science. It's not over, I don't mean that, but it's long enough that you have a sense of accomplishment. I suspect if you had it to do all over again, you would do it. That it's been a very rewarding life for you. Could you comment on whether it's true, or did you decide that you would have done something completely differently?
Well, as I indicated in the beginning, I feel as though much of my life has been directed by chance. If I hadn't been in a clinic for retarded children, I certainly wouldn't have gone looking at chromosomes in Oxford. So a whole area would not be opened. If when I came back to the University of Chicago, or firstly deciding to come to the university in '62, if I hadn't worked with Dr. Jacobson, I wouldn't be in hematology. So that I've often thought that if I write a book, and I don't intend to write a book, but the title of my book is Life as a Leaf on a Meandering Stream, because I think that summarizes what my life is. And I just meandered in a number of ways, and it's all been splendid. And so I wouldn't want to change it.
Sounds like evolution.
Or Candide, everything for the best in this best of all possible worlds. And with rare exceptions, I do feel extraordinarily fortunate.
Thank you so much for telling us about your stories.
Well, thank you for joining me in this, because it's been the two of us together.